To prevent bacterial diseases, K plus leak channels or K plus ion channels that is present on the bacteria is a promising target of our current research of drug discovery. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about the function of K plus ion channel or K plus leak channel that is present in the bacteria, or also known as KcsA channels. So basically KcsA channels or K plus leak channels or bacterial K plus ion channels, these are the channels that helps potassium ions to diffuse inside the bacterial cell membrane 10,000 times more readily than sodium ions. And by targeting those K plus ion channels or K plus leak channels, scientists actually develop a new drug to prevent various kinds of bacterial disorders. So in this video I am going to discuss about the mechanism of K plus ion channels. So how K plus ion channels or K plus leak channels actually works. So basically these K plus ion channels or K plus leak channels composed of several structures, that is, it has two peptides, that was M1 and M2. So M1 and M2, basically two peptides present in there, and there is a selectivity filter is there, selectivity filters which selects only potassium ions, not the sodium ions, because in selectivity filter there is a site for binding of a particular ion, and those K plus ions binds with this particular segment or peptide sequence of this selectivity filter, and they works with oxygen, and further they are selected by this selectivity filter. So whenever these potassium ions are outside of this plasma membrane, so basically this structure is present in the lipid bilayer, so we can consider this structure is a plasma membrane. Okay, so basically this is the plasma membrane structure. All of this structure is plasma membrane structure. Okay, plasma membrane structure. So, and here three peptides are there. One is outer helix peptide, that is the M1, another one is inner helix peptide, that is the M2, and another one is pore helix. So pore helix joins M1 and M2, that is the outer helix and inner helix. So basically selectivity filter is present between these two inner leaflets or inner helix structures, and it has basically 4 K plus binding pockets. So it has basically 4 K plus binding pockets, but at a time only 2 are occupied by 2 K plus ions. So at a single time only 2 sites occupied at the same time. So whenever this selectivity filter has 4 K plus selected channels or selected sites, that is specifically binds with K plus or potassium ions, but at a single time this selectivity filter actually binds with 2 potassium ions. Okay, so whenever a third K plus is insert into this selectivity filter, so basically whenever, so basically 2 is occupied, so whenever a third K plus is inserted into this K plus selectivity filter, there is a electrostatic repulsion occur, and those electrostatic repulsion creates a force. That force is actually helps this third K plus or first K plus detach from this second one, and it basically moves towards this side and ultimately moves from this opposite, exit from this opposite end. So this K plus exit from this opposite ends. So whenever this third K plus is enters into the selectivity filter, so this is the third K plus. Okay, so whenever this third K plus enters into this selectivity filter, number 1 and number 2 K plus are still there, and there is a electrostatic repulsion occurs, and those electrostatic repulsion helps to detach these 2 K plus ions, and by which a huge force is created, and by which 1 K plus exit from inner side. And Thus, this selectivity filter only allows K plus ion, not sodium ions. Okay, so only potassium ions can move through this selectivity filter. As because selectivity filter contains conserved pentapeptide sequence, and those pentapeptide sequences are glycine, tyrosine, glycine, valine and threonine. So basically these 5 amino acid, glycine, tyrosine, glycine, valine and threonine, present inside of this selectivity filter, and they particularly responsible for binding off with K plus ions. Okay, K plus ions. So basically this pentapeptide binds with K plus ion. This selectivity filter has 4 K plus binding sites, but at a single time only 2 is occupied, and whenever the third K plus inserted into this selectivity filter, those selectivity filter creates the electrostatic repulsion between first and second potassium ions, and by which a huge force is applied, and by this force those K plus ions actually inserted or exit from this selectivity filter and comes into the cytosol. Okay, cytosol or cytoplasm. So this selectivity filter allows K plus ion 10,000 times more radially than the sodium ions. Now scientists target these bacterial K plus ion channels or K plus leak channels to prevent cancer or to fight with cancer, various heart disease and other diseases. So if you like that video, please hit the like button. Don't forget to subscribe, and thanks for watching.